The U.S. has a really bad record of using herbicides and pesticides. Many herbicides that has been banned in other parts of the world are still heavily used in the United States.
We're very focused on the potential problems that the pollution of these waters can have on not only the aquatic ecosystem. If we're consuming that product and they're being affected by these pollutants that are entering the aquatic ecosystem as runoff, if we're showing that there is problems in these organisms in ways by growth, physiological effects, energy levels, whatever, what effect could that be having on us because we're consuming them? So I think it's a very holistic, ecology-driven issue that we're trying to sort of address.
I think that we have a very, very developed chemical industry in which they profit from manufacturing of herbicides and pesticides and fertilizers and genetic engineers organisms, and they have a very potent lobby. So the hope is that by doing more and more research and trying to provide more and more pieces of evidence, at some point the evidence will start bearing weight, and maybe we will change our political decisions to stop using these chemicals and to practice environmental responsible practices, which would be decreasing the dependency on these chemicals in our agriculture production.
Awareness is the biggest thing. Just acknowledging that a pollution or exposure to this sort of pollution of its kind does affect an aquatic ecosystem and a specific organism in a certain way, and that way is what we're kind of trying to determine, how is it affecting this organism. And I think having this hard data may bring about some awareness that there might be some other options to going about weeding or gardening. Using these pollutants may not be the best way to go about doing this.
The project is a collaborative project with the University of Buenos Aires. And what we are studying is the effect of herbicides on different species of crayfish that are commercially important both here in the United States, this is the Louisiana crayfish, and the crayfish that's farmed in Argentina.
We are currently working on Cerax quadricarinatus, and Dr. Vatnick is currently working on Procambarus clarki. And these species are biologically related. They have several common features from a biological point of view, and so it is interesting for us and for us, Dr. Vatnick and me, to know if these common herbicides applied both in Argentina and United States cause similar effects or different effects in order to generalize our conclusions.
We do typical exposures which an animal would be exposed to in the growing season, which the herbicides are applied, so it's kind of trying to mimic some kind of environmental conditions.
I have one control group and 3 treatments. I set up a plastic cup with 500 milliliters of dechlorinated water, which is kept at approximately 26-27 degrees. I also have an aeration system that, like in fish tanks, oxygen. So the crayfishes are individualized, one per each cup with oxygen, with a piece of elodea, which is an aquatic plant, and also are fed Monday through Friday with a formulated mixture food. Also, they are chronically exposed to sublethal concentrations of atrazine, low, medium, and high. So I clean the cups twice a week, Monday and Thursday or Tuesday and Friday. In that time, what I have to do is change the water, make sure the recipient is clean, and when the water is clean, I use a multi-pet to starting from a stock solution. I take small aliquots of the stock solution and I prepare the concentrations according to the volume I have. So another thing I do is every week I weigh every subject individually, and I have a record of during the month how much they grow every week, and I calculate their weight gain. The weight gain is a percentual amount of how much they grow, but it is independent of their initial weight. And with that numbers, then I do statistical analysis to see if there are significant differences between the weight gain between control group and the different treatments, the different concentrations of exposure of atrazine. And then, based on a preliminary study that I did last year, I was able to figure out the concentrations of atrazine that I use to be sublethal. So the objective is not that the animals die, just to see how much it affects their ability to grow. So I record that with the weight gain, and then my co-director Laura, she looks at them under magnifying glass, and she's able to tell me if they are male or female or intersex. They can be intersex, have both reproductive system, both gonads, male and female. Then she extracts the animal's liver and pancreas, and also I take out muscle to later on find out if the weight gain is affected. Maybe if I can relate that to the amount of energy reserves that the crayfishes hold in their abdomen, which is the little tail.
Nosotros hoy terminamos a disectar a los animales de esto y vamos a tener resultados. Hicimos metabolismo, vamos a tener resultados de todas las reservas. Entonces, la pregunta que queda pendiente es, si lanzamos el experimento que viene, ¿qué peso hacemos?
Ah, bueno, como para ver el peso que va a alargar esto Cami.
Sí, ella va a alargar un peso mucho menor. Yo creo que estos animales que estamos terminando tienen entre 100 y 300 miligramos. We expected to see this kind of effects that we see, increased mortality, reduced growth. We have not collected enough data to actually put together a big picture. I think that we will have to conduct several more experiments before we can see really the big picture. You know, obviously one experiment or one lab doesn't change policy. There are hundreds of studies that are being passed. So what I hope is that we just contribute to that, and at some point we get to a tipping point, which the consequences will be so clear, and the consequences will be so severe, and there will be so much documentation that at some point environmental considerations will outweigh the other considerations, in which different decisions will be made.